All right, found it. Okay, Dr. Miller, come on up. Thank you so much.
I haven't even done anything yet. But thank you very much to the group at Mass General for inviting me today to give a talk. And I'm really looking forward to talking to you a little bit about, my topic is also related to genetics. I'm a geneticist by trade at Boston Children's Hospital in the NF clinic. And the title of my talk is How Reading a Tumor's DNA Can Help Patients. So there will be some similar themes to what you heard about from Dr. Plotkin. And so I hope it doesn't seem redundant, but I'm just going to go through a little bit of like how I think about this topic. So I like to think of genes as like a library book because genes have important information in them and they're like the instructions how to do different things in the body. And also, as Dr. Plotkin was saying, they are spelled out in letters of DNA, and there can be spelling changes, and those spelling changes can lead to certain problems. Okay, so that spirally thing there that you see on the next to the book, that's the molecule of DNA. And then we think of chromosomes as kind of like a shelf of library books because the genes are lined up on the chromosomes. And Dr. Plotkin was already talking about how there's, a couple different chromosomes, one that's for NF1 and one that's for NF2 related schwannomatosis. So the genome is kind of like your whole library. And there are over 20,000 genes in the human library, 3 billion letters of DNA information. And it's very complicated to have everything work properly in the body. There's all different kinds of examples, thousands of different genetic conditions that are caused by spelling changes in our DNA. And it just happens to be that the ones that are most important for this audience are related to NF1 and NF2 schwannomatosis. So Dr. Plotkin already talked a little bit about how people who have NF1 or NF2 related schwannomatosis have already in their body one copy of the gene that has a problem in it. So the red thing, the red explosion there is meanings like that one of the genes is broken or not working properly. Okay. And sometimes that can happen in every cell in the body, and sometimes it can happen in a portion of the cells. And I'm going to show you a little image on the next slide that tries to explain that. Okay. So the big difference is you sort of have the baseline, whether in your body there's already one broken copy of the gene. And then later on, there can be something that happens to the other copy of the gene. And that's what can cause the individual complications in a certain part of the body. And I'll show you what I mean by that with kind of a diagram. So this is what would be typical for a person with NF1. And the blue color means that there's one copy of the NF1 gene that's working and one copy that's broken in every cell in the body. So analogous to this picture of the chromosomes where you have one broken gene. Okay. And that may or may not cause various symptoms. If it's just that pattern all through the body, you wouldn't have any neurofibromas, for example. For a person with the NF2 related schwannomatosis, there's a couple different things that can happen, and that I'm not sure how well it shows up. I think it shows up better actually up there than it does on my screen. That you can either have it similar to a person with NF1, which is on the left, where every cell in the body has one broken copy and one working copy of the gene. Or you can have it kind of on the right, where it's kind of we call it a mosaic pattern. In some of the cells, both copies of the gene are fine. In some of the cells, one copy is broken. Okay, and that happens more often with the NF2 related schwannomatosis. It's not super important in terms of the formation of different types of tumors, but it's just to give you an idea that sometimes it can be a little harder to find the genetic variant because it's not present in every cell in the body. And so I was saying that I was going to be a little bit redundant of what Dr. Plotkin says, but sometimes that's good actually to hear things a couple different times so they really have a chance to sink in. So show of hands, true or false? The NF1 gene and the NF2 gene are tumor suppressor genes. How many people think that's true? Okay, how many people think that's false? And how many people think, I'm not sure? Okay, all right, so it's okay, because you're not supposed to be an expert, but this is going to be a good opportunity. Now I know that it's good to talk about this even a little bit more. So the answer is true. They both work as these tumor suppressor genes. So their normal job, like Dr. Plotkin was saying, it's kind of like you can think of a tumor suppressor gene is supposed to slow down or keep the cells from growing too much, like the brakes on a car, okay? And I like to think of it like there's two sets of brakes. There's the regular foot brake, there's the hand brake, okay? So that's like the two copies of the gene. With either one of these conditions, as I was showing you, you could be born with already like your emergency brake is not working. That's like your one copy of the gene. The other copy of the gene that's working is like the foot brake, okay? But in any one part of the body, if you lose the foot brake, well, now the car is kind of just moving along and you can't slow it down. And that's analogous to if cells are growing in a certain part of the body where you can't slow them down. That's really what makes a tumor happen, okay? So in these individual areas that are marked red, that's where we've lost both copies of the brakes or both copies of the tumor suppressor gene, a gene that normally the job of that gene is to slow down or prevent the growth of a tumor, okay? And then I've just labeled these red dots with different types of tumors that could happen in a person with NF1, but they don't always happen, okay? It's only if you look at the chromosome picture, you got your one broken copy that you were born with, and then another broken copy happens in the areas with the red spots. It's actually what causes cafe au lait spots also, is that you've lost both copies of the gene in a little patch of skin, or it could cause an optic glioma or a plexiform neurofibroma or other physical differences that we see that are just isolated to one part of the body, okay? So yes, it is a tumor suppressor gene. Same thing can happen in people with NF2 related schwannomatosis. So if you lose both copies of the NF2 gene in the part of your ear where we see these vestibular schwannomas, that's what caused that to happen there. And if you're born with already one of your brakes is not working, then sometimes during your lifetime, the other copy of the brakes stops working also. Now the cells are growing out of control, okay? Another type of tumor that can happen is called a meningioma. And that's a tumor on the surface of the brain. Those things can happen in either people that are born with all the cells having a missing copy of the NF2 gene, or even in the person who has the mosaic form, okay? So I'm going to focus a little bit. I'm going to give you a few examples of things, and I'm going to focus first on the plexiform neurofibromas that happen in people with NF1. So one thing about these different tumors is they can be, anything that's growing is a tumor. It doesn't mean it's a malignant tumor. It could be a benign tumor. And a plexiform neurofibroma is a type of benign tumor. It's extra cells that are growing. And it's up to about 50% of people with NF1 have one of these. And they don't always know it because it might be on the outside or it might be on the inside, and they didn't know that they have it. And as Dr. Plotkin was saying, it comes from a particular type of cells called the Schwann cells. So now you see how these these two disorders are connected because they both involve Schwann cells. And what I want to talk about now is that through genetic changes, we already talked about the genetic change that's like in the NF1 gene. So if you lose both copies of the NF1 gene in these Schwann cells in a certain part of your body, you get a plexiform neurofibroma, which we called benign. So that's why I labeled it green here. So this person has a plexiform neurofibroma on their shoulder, let's say. Sometime in their lifetime, they could also get other genetic changes that happen within that tumor that could give it additional properties to keep growing and become a more aggressive or malignant type of tumor. And that's what we call a malignant nerve sheath tumor. And that's what's happening, why I labeled it red on the right side. So we're talking about there's the possibility that during a person's lifetime, a plexiform neurofibroma could turn into one of these more aggressive tumors. That risk is about 10% to happen, okay? Sometimes we don't find out about it until the tumor is more aggressive, and then it's harder to treat. So what we'd like to do is be able to use our knowledge of genetics to find these things earlier and maybe treat them more effectively. And that's what I'm going to talk to you about. One of the research projects that I've been involved with for the last 5 years, which was to learn about the genetic changes that are involved in that process of going from a benign tumor to a more aggressive tumor. Okay, so it can happen kind of like a traffic light. First, you have a benign tumor, it's green, everything's okay. It can turn into a sort of in-between tumor that's not quite as bad, but it's worse than the green one. That's the yellow one. And then it can turn red, okay? And what I have here in the diagram at the bottom is kind of like an activity map of the genome. Remember I talked about the genome as like a shelf of all your library books? So think of that from left to right. That's like all the sort of a diagram of all the genes lined up left to right. And there's two colors there. There's black and gold. And in the normal genome, or even in a benign tumor like the green tumors, the plexiform neurofibromas, everything's nice and quiet. It's done at baseline, okay? And what we see when we analyzed, we got 100 of these tumors together, which took a lot of effort, a lot of collaboration, including collaboration from our colleagues here at Mass General. And we analyzed those tumors with modern techniques of genomics to look at what was the genomic makeup? What did the library look like in these malignant tumors? And we found that it was very disorganized, okay? You don't really need to know all the details of that, but you can see when you look at my picture of the black and gold activity lines, there's a big difference between the normal tissue and the tumor tissue. And what all of that is, you can think of that kind of like, you know, when they talk about an earthquake and they have those things that measure the earthquake and it's supposed to be quiet and then all of a sudden it's all shaky and everything's going crazy. That's what's happening to the genomic information in one of these tumors. It's on every chromosome, there's big differences. And we think that those big differences are what can make the tumor more aggressive. But we can also use this pattern as kind of like a DNA fingerprint to identify the presence of that tumor. And so what we're going to work on now, and that's not just my group, but also there are groups now that are funded through CTF, a group from the National Cancer Institute and from Washington University in St. Louis, where we're going to be trying to demonstrate whether we can collect blood samples, like just when you go for a blood draw, for a blood test, they take it out of your arm, and they can find that if the tumor is starting to have this crazy pattern where the genes are getting all disrupted, some of that DNA from the tumor is going to be floating around in the person's bloodstream. And we can use very sensitive techniques to detect the presence of it in the bloodstream and look for that DNA fingerprint. And we're hopeful that we might be able to find that DNA fingerprint in the blood even before a person has the symptoms that they know that there's a bad type of tumor growing in their body. And then we could do an imaging test and try to find where it's located and perhaps be able to, provide some treatment at a sooner or earlier stage that will be more effective. And we call this type of early detection of a tumor in the blood, we call that a liquid biopsy, because we're not actually taking a sample directly from the tumor. We're just finding evidence of the tumor in the person's blood. Okay. So that is example number one of how we can read a tumor's DNA to get information that might be helpful in treatment. This is going to be taking a few years to really be able to see whether this works or not, but we're hopeful that that's going to help people with NF1. It possibly could be used for NF2 or any other type of tumor. I want to give you another example of how reading the DNA can help with learning about how to treat tumors. This is based on the work of my colleague, Mariella Philbin, who's at Boston Children's Hospital and Dana-Farber Cancer Center. And what she's working on is a different type of tumor that can happen in both people with NF1 and rarely, but can happen in people with NF2 related schwannomatosis, which is called a glioma. It's a type of tumor that starts as a benign tumor, but it could become more aggressive also. And there's some nice color pictures here. Basically, what I want you to know about this picture is each of those different colored things in the picture is a type of cells that are in the brain. That's where these type of tumors happen. And there are healthy cells and also different types of cells, like there are neurons. There are also support cells for the neurons, and there's a mixture. But once these tumors become more aggressive, the pattern of what they look like under the microscope is different. There becomes more of these tumor cells, and they can sort of wall themselves off and start to grow, and the body's defense mechanisms can't get to them. So what Dr. Philbin's doing is trying to learn more about how the tumor cells interact with the normal tissue so that we might be able to develop better treatment. And what she does in her laboratory research is to apply DNA sequencing, which basically means reading the letters in the books to try to find out what's different about the cells that are in these tumors. And rather than just taking a whole mixture of different cells and trying to read their books, because that would be confusing because you're reading multiple types of cells at once, she does a thing where they separate the cells out into individual compartments. So there's like a tray with little wells in it, and you get like one cell to go into each well, and then you can read the library that's in each cell separately, so it's not overlapping with the signal from the other cells. And then you can try to learn about the types of cells that are in the tumor and the different proportions of cells, what proportion of normal cells, what proportion of tumor cells do you have there. And you can also learn about, when you sort out these cells, you can actually learn about how they talk to each other. So you can sort of see how they're organized by reading the sequences of the library of each cell. You can categorize them into the categories of tumor cells and normal cells. And you can also see what genes are being expressed by the normal cells. And those genes are actually signals that are sent from one cell to another. And you can also separately see what signals are being made by the tumor cells that are being sent back to the normal cells. If you had these all mixed up together and you were reading sequences, you wouldn't know if those signals were coming from the normal cells or the tumor cells. So it's the process of separating them out and doing single cell sequencing that helps us get a better understanding of what's going on within the tumors. And there's a whole lot of information. There's a whole lot of research that goes along with it that I'm not going to have time to talk about. But the the take-home message is that we're using single cell sequencing to learn about tumors. And if we can find out what the individual signals are that are making those tumors grow, that provides a chance to target those signals with a specific treatment, like a molecule that would block that signal. Okay, so that's really the goal of how we're trying to help patients with single cell sequencing. And then the last thing that I'll talk about, example number 3 of how reading a tumor's DNA could help patients, is an example related to NF2 related schwannomatosis. So I mentioned before that in addition to vestibular schwannomas, the most common type of tumor in people with NF2 schwannomatosis, they can get a meningioma, which is a type of tumor on the surface of the brain, which I marked in yellow here. So in that yellow tumor, it's the same phenomenon. There was already one copy of the NF2 gene that was broken, and then the other copy got broken. So now there are no breaks on the cells and the cells are growing. And researchers have been, just like we did with our MPNST tumor consortium, there's a group of researchers led by a group at the University of California, San Francisco, where they have been studying these meningiomas. They've collected over 500 of them and read the sequences of their DNA to figure out what controls the growth of those tumors. And it's not just that the NF2 gene is broken in both copies. That's that's the start of the process. But just like with the MPNST tumors, where there have to be other genetic changes that happen to keep the process moving, one of the things that happens in these kind of tumors is it's not actually a change in the DNA sequence, it's a chemical modification of the chromosomes called methylation. And what I'm trying to show you here is that this is a chromosome with a copy of, say, the NF2 gene on it, all right? And the normal thing that's supposed to happen with that gene is it can make more copies of itself. So all those little green tick marks there are when the gene is working properly, it's making copies of itself, and those copies of the gene are doing their job to slow down the growth of a tumor, okay? That's when the gene is working normally. In a tumor cell, there's this methylation can block the gene from working. So even if it's the NF2 gene or some other tumor suppressor gene, the methylation can block it from working. So now it's like turning off the brakes on the NF2 gene, and these methylation signals can be placed down in other parts of the genome and turn off other genes as well. So like I was saying, you start with the NF2 gene being the beginning of the problem, and now other genes are getting turned off by this process we call methylation. And what these researchers showed is that in the meningiomas, there were different patterns of the way genes are turned off depending on the type of meningioma. And there were basically, instead of them all being the same type of tumor, they found that the meningiomas are actually like 3 different types, depending on the pattern of this methylation. And they could also see that those patterns of methylation, those categories, correlated with how the tumors behaved. So I won't go into too much detail, but for example, one group of tumors, just even though they look the same under the microscope, the way that their tumor suppressor genes were turned off made them more aggressive tumors that had a worse outcome for patients. Okay, so they also used that information though. So when they looked at the tumors that were more aggressive, they could see by the methylation pattern which genes were being turned off. And then they thought about it and they said, well, if we know which genes are being turned off, we're going to choose a treatment that addresses that particular pattern of genes. And so they did that in some model systems, and they tried drugs that were specifically focused on the genes that were affected by that pattern of methylation. And in the model systems, they showed some improvement in being able to treat the tumors. So that's going to be like a next step, is to try to translate back to taking care of patients that way. So I'll just summarize, because I think I'm out of time, that the main take-home points are that you can have the baseline genetic changes in either the NF1 gene or the NF2 schwannomatosis genes that can cause the benign tumor to form. It would then be additional genetic changes needed to make them into a more aggressive tumor. And that I've showed you 3 examples of how we're understanding the genetic information to provide some ways to help patients either with early detection or trying to select better treatments. So thank you very much for listening, and feel free to ask questions.